4 hours.
Okay, a couple of other administrative things. First of all, as I told you last time, this class is supposedly being audio cast, but it's not being webcast. Last time, not at this end, but at the other end, the audio cast screwed up, and that happens. So, it's a good idea. I recommend strongly coming to class. However, because we don't have the video cast, I asked the people who do this stuff to please put up last year's video cast for the whole quarter. They're not going to be exactly the same, but they'll be pretty similar. Those just went up this morning. I haven't even looked to see how they're organized. They may actually be flooded. That may be, if you look on the site, that's all you can find now. They may be flooding the thing. I have to organize them, and I'll do that later today. And I will also, in a day or so, give you a list of which of the correspondence between the syllabus items and the dates on the, or however, or the numbers or whatever they are on the last year's video cast. So I'll set that up in the next day or so. So if you miss a lecture, you can do that. I don't really recommend using that as a sole basis for getting all your information. But if you miss something or want to hear something over again, that's the way to do it. Another thing I'm experimenting with, and I'm doing it today actually, but only halfway, is there's a program called Camtasia, which is basically a screen capture program, and that's what this microphone is for. And so this whole lecture will, in fact, go onto my computer, and I'm going to try to put it up on YouTube. Whether that's going to work or not remains to be seen. Okay, so this is an experiment. I would certainly not count on using it, but we'll see what happens. So I'm going to try doing that with all the lectures, but we'll just see. I think that's the only announcement I have to make right now. So let's get on with our actual lecturing. Last time we were talking about the fact that, at the end, the effects of genes are pleiotropic. Every gene affects many traits, and every trait is the product of many genes. And we illustrated that just at the very end of the hour by these fruit flies, each of which has a gene for a different eye color. There's the normal one there, but this is more red. These are kind of actually white, although the picture doesn't show up very well. The notable thing about this is that although each of these it has a one gene, each of these different flies has a one gene mutation that has an effect on its eye color, all of the mutants have abnormal viability. They don't respond well to stress, etc., etc. They have other difficulties besides the eye color thing. This is evidence for the fact that genes almost always have multiple effects, even though we often name them for some conspicuous effect like eye color that we can see. We'll talk a little bit more about that too later on. Another example of the pleiotropic effect of genes, which is one that I'm rather fond of. This is something we're going to go into in more detail later in the course, but there is one particular kind of receptor on nerve cells called an NMDA receptor, and you don't need to remember that at this point. Later on you will, but not now. What this NMDA receptor does is when the cell gets certain sorts of input from other nerve cells, this NMDA receptor opens up a channel to the interior of the nervous cell that specifically lets in calcium, or calcium ions, from the outside. Now, that's uniform. That's true throughout the whole nervous system. That's the single basic effect of the NMDA receptor, and therefore, that's the important thing about the gene that makes this NMDA receptor, which is a protein. However, what's interesting, and the pleiotropic aspect of the thing, is this. For neurons in the spinal cord, nerve cells in the spinal cord, the role of that calcium coming in response to the stimulation of this NMDA receptor is that it's crucial for generating the alternating steps of bipedal or quadrupedal locomotion. In the certain parts of the brain, those very same NMDA receptors, which are made on the very same genes, the calcium that comes in when they're activated plays an absolutely crucial role in learning. So obviously, learning and walking are two totally different things, but this one gene is needed for both. Okay, so that's a really good example of the pleiotropic effect of genes. So I'm just rubbing it in. What do we say here? Again, every gene affects many traits. Every trait is a product of many genes. So don't forget that. Moreover, if some gene is changed, is mutated, it will have multiple effects on the body, not just one. I guess we've said it 5 different times, 5 different ways. Let's move on. Now, so the pleiotropic effect of genes is one absolutely fundamental thing about them. Another absolutely fundamental thing about them is that genes provide information that is essential to building an animal. That we all know. They make the proteins, and the proteins have all sorts of roles and growth and so on and so forth. Enzymes, which are needed for all the chemical reactions, so they're absolutely crucial. But they can never be the whole story by a long shot. And the reason we know that is that every cell of a given individual's body, except the sex cells, all the other cells, have exactly the same genes. They are identical genetically. Every single cell in your body is the same as every other cell. The genes in every cell of your body is the same as the genes in every other cell of your body, and yet you have different kinds of cells in your body. You have liver cells, brain cells, skin cells, so on and so forth. There are different lines of cells. When they divide, they say skin cells, or cells can generate skin cells, can only generate skin cells. Ditto for liver cells, so on and so forth. And yet they all have the same genes. Well, the only way that can possibly be is if this common set of genes of an individual are somehow read out or expressed in a different way at different cell types in the body. Every cell of a given individual's body has the same genes. You have a liver cell is very different from a brain cell, and so on. So genes must get expressed differently in an individual's different types of cells. Okay. If genes are not the whole story for making up your body, one individual's body, then they're not going to be, making the cells and so on in the body, they're not going to be the same thing, they're not going to be the whole story for determining your traits, your psychological traits or your physical traits or anything else either. There's going to be something else besides the genes. There has to be. I mean, it's not just kind of, maybe it's so. It's absolutely got to be so. It's fundamental. There has to be something else that can influence the building of your body, because it wouldn't work otherwise. If the genes were the only thing, we'll see this in just a moment, you would just be a ball of cells, all identical cells rolling around on the floor, or something like that. So the question then, it's definitely the case. It has to be the case that different cells read out the genes, or utilize the genes, or express the genes in different ways. The question then is, what might one imagine could determine which genes get read out in a cell? There are lots of possibilities that over years of research were raised, some of which turned out to be righter and some of which turned out to be wronger. 3 ideas that have been raised many times, and all of which have some degree of validity to them, is one. Well, sorry, let's back up a bit. When we are all made, we start out as a fertilized egg, single cell. Next step is that divides into 2 cells. Next step, that divides into 4 cells, and so on. We have repeated cell division. Now, as I say, if all the genes were just read out uniformly in all these cells, we would just become a big ball, a very large version of that. Okay, that's obviously not what happens. Now, what things could cause different cells, to differentiate, to become different in different ways? What could cause differential gene readout? Well, one possibility is that it might be that the cytoplasm, the interior, the chemicals in the different parts of the fertilized egg are different from one another. And so when the cell divides, maybe this guy has somewhat different chemicals inside of it than this guy, and those different chemicals in the 2 different daughter cells cause the genes to be read out in those cells in a different way, and so on down the line. That's one possibility. Non-uniform factors from the original egg distributed differently into daughter cells, and so on. The second possibility is it might be that something changes over time. As the cells divide, maybe different genes get read out. And so maybe liver cells differentiate, they become created first, and so one kind of gene gets read out in them. Maybe skin cells rise later in the developmental program, and so maybe different genes get read out in them. That's another possibility. Third possibility is that once there's been a certain amount of development, you've got this egg in the mother's womb and so on, and eventually you get to a stage of something like this, and you get to a point where there's cells that are on the inside, there's cells on the outside that are more exposed to the mother's bodily fluids, and so on and so forth. It could be that different that cells that have different chemical and physical environments, that those environments trigger the reading out of genes differently in those cell types. So that's different cellular microenvironments might determine what genes get read out, and then that would be a process that would continue and get more exaggerated perhaps as development occurs. So these are three possibilities. It turns out all these kinds of things occur to some extent in some kinds of animals. But in vertebrates, which we are, and in particular in mammals, but in all vertebrates, this last one is especially important. It's the lion's share. And there's a whole field called experimental embryology, which has done probably hundreds of thousands of experiments that have shown this in various ways and worked out the details of it in various ways. Let me just give you a prototype for one of the kinds of experiments that make the point. This experiment, and the drawing is schematic, but basically this is the kind of experiment that's been done. In a developing amphibian embryo, there comes a point during development where there are cells at one place in the animal that are going to become skin. They're going to divide, and eventually they're going to turn into skin cells. And there are cells somewhere else that are going to become nervous systems. Okay? Now, here's the question. Suppose you took a little melon scooper and made a little scoop from there, and we have two melon scoopers, one on the left hand, one on the right hand. We make a little scoop, put one little scoop of cells from the center of that in one melon scooper, and a little scoop of cells from over there in the other one. We cross them and stick them back in the holes left by the other one. So we take some of the tissue from here and stick it over here, and some of the tissue from here and stick it over here. So now we have cells over here that were supposed to become, originally, if left to their own devices, would have become nervous system cells, but they're in a whole field of cells that are destined to become skin cells. What do they become? Do they become skin cells or nervous system cells? Interesting question. If you do this early enough, they become, their surround talks to them and tells them, no matter what you think you are, you're not. You are in this case, a skin cell. And vice versa over here, no matter what you think you are what the cells around you are, namely a nervous system cell, and that's what they become. There comes a point during development when they become specified. If you leave them alone before you do the crossing experiment, they become specified, and then if you cross them, they become what they were already thought they were. So it depends how you do the experiment. But if you do the experiment early enough, and this is the crucial thing for our point right now, if you do the experiment early enough, what they become determines what they're next to. So obviously the cells next to them must be somehow sending them a message or stimulating them in some way that causes them to become the same thing as their surroundings. It influences what genes get read out in them. So that's a very good illustration of the fact that the environment, the physical chemical environment of a cell can control what genes get read out. So that's what I just said, so there it is. So cellular environment controls gene expression and, of course, cellular development as a whole, and ultimately, therefore, development of traits. So genes alone never tell the whole story, I should say, of what a cell or organism, i.e. us, will become. So if what genes are expressed varies from one cell to another, then where is that control exercised? Proteins are made via steps. We have the gene, it gets transcribed into messenger RNA, and the messenger RNA gets translated into protein. So there are basically three ways that control could be exercised in principle. One is that whether or not transcription occurs could be controlled. A second is that whether or not translation occurs could be controlled. And the third is it might be that the protein's actions themselves could be controlled. Well, it turns out, again, all three things happen. All three things are important, but the most important is transcription. So we will focus on control of transcription. So far, the sort of grand scheme of development that this information I've given you sort of puts in place is the development's kind of a circular thing. Gene action affects the structure and properties of the developing organism, which, at any given point of time, the organism as it is influences the cellular environment of cells that are still developing, and those in turn affect which genes get expressed, and it goes around and around and around all throughout the course of development. So it's very much not a thing that the genes cause something, or for that matter, the environment caused something, or the cellular environment causes something. It's a circle. There's a continual process of interaction. We're going to elaborate that story now. This is a little bit more complicated than that. Oh, okay, right. I think now is the moment. So as I told you last time, I made the first of what I hope will be a short series of little video talks. This one's about half an hour long. And some important details about genes and the control of the transcription have been put into this first one. It's on the web page. There's a link to it on the course webpage, and now is the time to watch it. I mean, not this minute, but whenever you're ready. And I would do it within the next few days. So my lectures will not depend upon it, but your doing exams will definitely depend upon it. And again, I apologize. As I said yesterday, or said on Monday, this thing is not very polished. Hopefully I'll get better at it. But hopefully you'll be able to understand it. Okay, so we are finished now with the broad biological principles of development. And we're going to now apply those and see how they operate in the nervous system in particular. It turns out some special stuff goes on in the development of the nervous system that doesn't go on in the development of the rest of the body, at least not so much so. Okay, first of all, we just saw this experimental embryology experiment with the amphibian, with the skin, and the nerve cells, with one kind of cell influencing the cellular environment, influencing what a cell becomes. First thing I want to show you is that rule applies, of course it applies to the nervous system, because the nervous system is made up of cells. But more importantly, it affects the development of the wiring of the nervous system. Exactly the same principle, only this experiment is a little bit more complicated than the previous one. So first of all, I have to give you a little bit of background. Here is a normal developing amphibian. And we're in the visual system of this animal. Here is its retina on left side and right side. And the nerve cells, some of the nerve cells of the retina that project into the nerve cells of the retina that project into the brain, and we'll learn more about that much later on in the course. The cells of the retina that project into the brain project in a sort of screwy way. The cells in the front of the retina go to the back of the part of the brain that receives them. It's a place called the optic tectum, but I don't have that in the slide, and I don't want you to know it anyway. But the part of the brain that receives them, or at least one part of the brain that receives them, the cells in the front of the retina go to the back of this part of the brain, and the cells in the back of the retina go to the front part of the brain. Okay, that raises a question then. Suppose that during development you screw around with the eye so that you reverse it, so back is front and front is back, as indicated there. What happens? Is it still the case that the cells that are now in the front go to the back, or are the cells that were supposed to be in the front go to the back? Could be either way. Same story as with the previous example with skin and nerve cells. Well, it turns out that if you do this early enough, the current front of the eye goes to the back of the tectum. In other words, these cells that are in front are told probably by the eye cup which ones they are, front or back, and then they get their status gets changed and they project to the back. But if you wait past a certain critical time in development, the reverse is true. So initially the current front of the eye goes to the back of the septum, but if you do it later, if you do the reversal later, then the former front of the eye goes to the back of the septum. Again, it's this part of it that we're most concerned with. This idea that the cellular environment of a developing cell of a developing nerve cell can influence the neural connections it makes and therefore the circuitry of the nervous system. And of course, that's what we're all about, is the circuitry of our nervous system. So that's a very important fact. But it goes further than that. So, just like I said, just like the rest of the body, how the nervous system grows and wires up is greatly influenced by cellular environment. That's just saying the same thing again. However, but in the nervous system, a new dimension is possible. Because what happens is, you don't just sort of, the nervous system doesn't just sort of grow and grow and grow and get all hooked up and then start working. That's not how it happens. Very early on, after a certain amount of formation of nerve cells and so on, they start responding to stimuli and affecting other nerve cells, and we'll talk about the details of some of these things later. But they start being active, and the nervous system starts processing information and sending messages from one nerve cell that's already developed to another nerve cell that's either already developed or in the process of developing, throughout the whole process, or throughout much of the process, all the later process at least, of development of the embryo. Therefore, a new thing is possible. Maybe event-caused activity. This baby is developing in the womb, but it's not as though there's temperature cues, there's the mother bouncing around, there's probably cues from that. There's all sorts of stimulation that it's getting. The mother is doing things, and that's changing her hormonal, the chemical environment of these cells, all sorts of things going on. Wherein events in the outside world, the activities of the mother, and maybe even to some extent the activities of the developing embryo, influence which nerve cells which nerve cells that are already developed fire, become active, and this in turn could influence the further development of cells and circuitry that is not yet, that's still in the process of developing. So, and this just says this again, maybe event-caused activity of nerve cells in developing nervous system influences the environment of other growing nerve cell and so shapes further development of neural circuitry. Interesting possibility, obviously greatly complicating the picture of what goes on, potentially greatly complicating the picture of what goes on in development. Now, I wouldn't be telling you this if there were not some validity to it. It actually happens. And what I want to do next is give you 3 examples of evidence that it happens. And these examples are going to start with something that's very directly neural, having to do with neural circuitry. Then we'll move to something that has to do with reflexes and sort of rudimentary behavior. And then finally we'll talk about something that's even a little more sort of kind of learning. We'll see what I mean in a minute. So first example, the one that's very neural in nature. Again, first of all, I have to give you a little bit of background. Here are the 2 eyes, the left eye and the right eye of a young of a neonate. In this case, it might be the neonate of a cat or a dog or you, it doesn't really matter. Now, suppose this young organism sees this cat. An image of the cat gets projected on both retinas, 2 eyes. 2 projections are not exactly the same. They're a little out of kilter. There's one seeing it from over here, and one seeing it from over here, and one seeing it from over here. So they're slightly different from one another. Now, the cells of the eye, one particular group of the cells of the eye, project into the brain. And they actually go to a part of the brain called the thalamus, which I don't care about. We'll talk about that one later on when we talk about vision. But from the thalamus, they go on to the back of the brain here on the cortex, the visual cortex. And so that's what I want to focus on. if you look in a normal animal at any particular visual cortical cell, what you find is that each cell, roughly speaking, receives input, or usually receives input that's derived from both eyes, and typically from the corresponding part of the retina on both eyes. So they look at it over here, or they look at it over here. So each cell gets input from both eyes and from the corresponding part of both eyes, each cell in the visual cortex. That's the normal situation. And one can see that over here in this graph. What this is a graph of is the percentage of neurons in this particular layer of the cortex which get input only from the left, only from the right, or various intermediate amounts. And what you see is most cells get input from both the left eye and the right eye. That's the normal situation. However, suppose you were born cross-eyed, or suppose a fiendish neuroscientist did surgery on you, and this would be done on a cat, typically not a person, in which they screw things up a little bit, so your two eyes weren't quite in register with one another. In us, we call this being cross-eyed. If you're born that way and nothing is done about it, then what happens is you grow up with the two corresponding parts of the retina not seeing the same thing, even remotely the same thing. And it turns out if you do that, then instead of getting a picture like this, you get a picture like this, where all the cells, almost all the cells, are monocular. Each cell gets input either from the left eye or the right eye, but not both. And what that does is it totally screws up the individual stereoscopic vision. Okay? Now, this is understood, and people that are cross-eyed wear special glasses to bring things into register, so this doesn't happen now in most cases. But the importance of it from our point of view is it's very clear evidence that the development of the wiring of the visual system depends upon what the individual, the young individual, the neonate, is seeing soon after it's born. So that's the most clear-cut example, and it's been studied a great deal. Most clear-cut example of this general principle that I'm telling you, that the activity of nerve cells during development, because development is still going on for sure, postnatal, early postnatal, the activity of nerve cells and events occurring can influence the development of the circuitry of the nervous system. Now the second example, is a little bit, it's not directly neural. You have to sort of, but it's obvious that it's something is going on in the nervous system. When fairly soon after a young kitten is born, it will have three absolutely standard visual reflexes. One of these is the so-called visual blink. Actually, with these glasses on. If you have a pane of glass in front of the animal's face, and you go like that, it blinks. No surprise, right? Because even though the glass prevents any wind currents or anything from reaching the face, just as a visual reflex, this big looming thing coming toward the eye causes the animal to blink. That's called the blink reflex, and it's obvious that it makes sense. We all know it's there. A second thing that happens is called the visual placing reaction, and this you may or may not be aware of. You take a young kitten a week or two after it's born, and you hold it up, pick it up, and you bring it up to an edge, just exactly like this. So here are the front paws, and you hold the animal so its paws are a little below the level of the table. You bring it up, and it's looking at this. When it sees the edge of the table, it picks up the paws and puts it on the table. A lot of you, if you're a cat lover, you've probably seen that. That's called the visual placing reaction. And all self-respecting cats have that a week or so after birth. The third example is called the visual cliff. Actually, I could demonstrate that over here. Here's a small visual cliff. You put the cat on a little ledge, so on one side there's a bigger drop than that, and on the other side there's just a little drop, maybe one like that. Now the cat's all confined, it doesn't want to stay on this little thing, so it's going to step off, right? Which way does it go? The big drop or the little drop? It always goes to the little drop. Even though cats don't mind a bit of a fall, it goes to the shallow side, and that's called the visual cliff reaction. The young cat, sees this drop and prefers to go to the shallower drop than to the bigger drop. Okay, now those are all highly adaptive reflexes. And one might think that they would all be totally inborn based upon the circuitry for those reflexes would all be fully developed when the animal was born. Not so. And one can show that it is not so by, well, one way one can show it's not so is if you cover the animal's eyes during the first week or so after birth and then test these things. None of them work. So clearly some sort of input was required, but it's more subtle than that. Very particular kind of input was required, and it's shown in this experiment. Here what we have are two kittens. One of them is being allowed to walk around under his own steam in the circular arena. By the way, these are stripes on the side of the arena. The dots are an artifact of the scanner that I used to make the picture. They're not really there. They're really there, but they weren't in the original. As this guy here, this free to walk around, walks around, the world moves past him. These stripes move relative to his retina, based upon his or her own locomotion around the circular pen. The other kitten is seeing the same thing, but it's in a little carriage. So whenever this guy moves forward, whenever this guy moves this way, this guy's carried this way. He sees the same thing, but it's not under his own control. What happens is this guy develops these three reactions. This guy does not. So it's not just the visual stimulation per se. It's more subtle than that. It's the interaction between, it's visual stimulation of a certain kind that is produced as a result of the animal's own voluntary movements. And that's what's needed to get these three things to finish their development. Obviously, much of the nervous system, much of the neural circuitry for them was already there, but for that circuitry to be finished off and become functional, the animal has to have this particular kind of experience of seeing the world slip past its retina as a result of its own movements. The last example I want to give you is a phenomenon called imprint, which some of you may have heard of before. This occurs in various ways in pretty much all animals, but where it's been most studied is in certain kinds of birds, and ducks, for example. And if you look, well, you've all probably seen a little group of baby ducks following its mother on a pond or a stream. The mother goes and they're all following behind. That following reaction isn't actually inborn. It has to develop. And an interesting question is, what does the duck follow? Obviously, the duck follows its mother, right? At least it follows another duck. Well, not necessarily so. That's what would seem to make biological sense. You might think that as part of the development of the organism, that the neural circuitry would be set up in such a way that the stupid little duck would know the difference between a duck and a dog or a cat or something like that. But it doesn't. All right? Turns out that what the little baby duck comes to follow depends upon anything that moves and is making noise at a certain critical period after its birth. And this is a graph of that sensitivity. Various days, or just hours post-hatching, and there's a big peak here at around 16 hours, and it falls off on either side of that. At a certain critical period during this animal's development, the animal or the duck will learn to follow whatever it is it sees. And if it's usually, of course, in real life, it ends up being the mother duck, because the mother duck's staying around her children. But if you, as an experimentalist, go in and let it be a barking dog or yourself or a ball that makes noise when it bounces, it doesn't matter what it is, that's what the baby duck will become imprinted on. And not only will that be the thing then that thereafter the baby duck will follow, and not other things, only that. Moreover, when the baby, this part of it's a little bit apocryphal, there's disagreement about whether this is true or not, but as far as I'm concerned, it's true. When the duck grows up, the thing it thinks is suitable to mate with is whatever kind of thing, not the specific individual, but the kind of thing that it saw during that critical period. What's happening is during that period, the duck is learning what a duck looks like, except if you fool it and put something else in there, it learns whatever it was you stuck in there. This is the phenomenon of imprinting. And this last bit about the sexual proclivities of the duck are, I'm not sure whether this has held up. There's been some controversy. I'm actually not sure, but it's a good story. So, for the purposes of exams, it's true. All right. Okay, so this obviously is another case where aspects, it's a kind of learning, of course, but it's a learning that only occurs at a very, very, very precise point of time, so-called critical period learning. And when any learning that occurs, obviously involves some change in the nervous system, and changing the wiring of the nervous system. And so this is another piece of evidence that the wiring of the nervous system can, not only during prenatal development, also during early postnatal development, can be influenced by events occurring in the animal's life. So we have to elaborate our picture of the scheme of development. It is this kind of circle that we talked about, but there's another thing in here. And what we stick in is interactions with the external world. So the nervous system makes use of information in the outside world to help direct its own construction. And this happens because the nervous system doesn't just develop and then start working. It's working through a large portion of the course of the developmental program. So this is a very good picture of what development in all vertebrate animals, and certainly of ourselves, is like. So it's very complicated. And as we see, and especially as you see when you see the video that you're going to see, it's all very complicated. Okay, so that's what I wanted to say about the application of the basic biological principles to the nervous system in particular. And I now want to turn to these questions that we raised at the beginning last time about the origins of the differences between us. Some of us are smart, some of us are stupid, less smart. No doubt there's nobody stupid in here. And so on. And the question is, why are there those differences between us? To what extent are they due to genetic differences between us? And to what extent are they due to rearing differences between us? Oh, okay. Remind me to remind you. So you have these questions, before I go into what we're talking about now. We have these questions that are on the website that are your first set of study questions, and you can I can start trying to do them any old time now. The way you're going to answer them is an answer sheet will be Scantron type answer sheet will be posted. And you will have it has to be filled out by a certain date, probably by next Thursday. I'll send you an email telling you exactly what the rules are going to be. Now, if you want to start doing it early, that's fine, because you can keep doing it over. You keep taking it over and over and over again, as long as you want, until this. And there may be a record of your progress, but I'll never see it. All I'm interested in is your final score when the thing closes. So you can take it and do it again as much as you want. When you take the thing, what will happen is, or what should happen if the program works right, which last year it didn't, but I think we got it fixed this year. What will happen is you will get, for each question, you'll be told your total score, and for each question you'll be told whether you got it right or wrong, but not what the right answer was. So the idea is that you'll then, those things you got wrong, you'll go back and think about it some more. Now obviously you can just keep taking the thing until you get it perfect by trial and error, but it's not going to do you any good at all. And since it is worth some credit, but very little, it's not an adaptive thing to do. The adaptive thing is to get them all right, know why you got them right, so that when you see a similar kind of question on an exam, you also do it right. So this is here not to evaluate you, this is here to help get you ready to take the exams and to help you learn the stuff. And so you should keep that in mind when you're doing it. So, and this answer sheet will appear pretty soon. I'll send you an email about it. It appeared briefly yesterday while I was setting anything up because I was not doing something right. And if you took the exam then, it won't matter because you're going to take it again anyway. But it's gone now, I think. Okay. So back to our question about the origin of differences between behavioral differences, trait differences, behavioral trait differences between one individual and another. Are they due to genetic differences, or are they due to environmental differences, or are they due to both? Okay, first of all, it's worth pointing out that we are all pretty darn similar to one another, even if there are differences between them genetically. Even though there are genetic differences between us, we are all pretty similar. not only are we pretty similar, in fact, if you look at, this is a phylogenetic tree of primates. Here are we, Homo. There is a chimpanzee. There is a gorilla. And the genetic similarity between us and a chimp is 98.77%. So genetically, we are very similar. Now that small difference is very important, but we are very similar. But so if these two guys are that similar, then presumably you two guys are even more, I presume, are more similar even than that. So we're very similar, but we're not identical. There are some differences, and such differences as there are between individuals have major impacts and cause inter-individual variability. So it's important to remember that we're all very, very similar genetically, but we are different, and those differences do have implications. And what we're trying to do right now is to understand the nature of those implications. Now, before we start doing that, and looking at specific psychological traits, I want to make a point that people, at this stage, as I give this lecture, everybody thinks they understand it, but when it comes to exam time, an awful lot of people don't. So, we've just spent most of this lecture and part of the last lecture arguing that all traits depend upon genes and cellular environment. Okay? And now we're saying, okay, to what extent do differences in intelligence, or whatever the trait is, between individuals depend upon genes or environment? And the reflex answer to that, based upon our emphasis that it's both, is that it's an unanswerable question. We shouldn't even be addressing. I mean, we can address it, because everything depends upon genes and environment. And that's absolutely true. Every trait depends in an inextricable way, that circle we showed, on genes and cellular environment. And indirectly, a lot of them depend upon external environment during early development. But no matter how much that's true, and it's absolutely totally true, that doesn't mean that if you have two individuals that are different from one another in their behavior or their traits, you can still ask the question as to what extent is that difference between them due to genetic differences between them or to nurturing differences between them. That's still a perfectly answerable question. It might be, the differences between them might be for entirely due to genetic differences, or it might be entirely due to upbringing differences, or it might be to some combination or interaction of the both, or some interaction between those things. So we can ask, are particular observed differences between two individuals due to gene differences or developmental environment differences? Perfectly answerable question. I'm emphasizing that because every time I give an exam asking that in some form, I only get about a 25% correct rate on the answer. I hope that I don't stress it quite so much as I am this time, so hopefully that won't happen this time. Whoops. Oh, that's saying the same thing. You can look at the slide later, but I don't. That's why when I light, I don't want to focus on it. Okay, let's start talking about the nitty-gritty. Now, there's a lot of data on the extent to which variation in various psychological traits is genetic variation between individuals, or environmental or upbringing, nurturing differences, or educational differences between the people. But the greatest amount of study has been on IQ. Now, I am not going to, the definition of IQ is an interesting and important topic, which this course is not going to go into. There are tests that have been devised that do a very good job of predicting how you'll do in school. And that's why they were devised. And they are called IQ tests. Whether there really is something called IQ or intelligence, it's an interesting and important issue, but it's not one that we're going to address in this class. So for our purposes, IQ is what the IQ test measures. Okay? I don't want to, that's not an issue I want to get into. I do want, but I'm going to refer to it not just as IQ score. I will talk about it. I use the word intelligence, but I probably shouldn't, strictly speaking, but I will. The question we do want to address is the extent to which differences in measured IQ, or loosely speaking, in quotes, in intelligence between one individual and another, are due to genetic differences between those individuals, or environmental differences, or educational differences between those two individuals. So let's start with some facts. If you look at the correlation, here's why I wanted you to all read that handout. If you look at the correlation in measured IQ between a biological parent and its child, it's about 0.48. That's a pretty good correlation. It's a highly statistically significant correlation. There's a huge amount of data on this. And it's a good correlation. Not, a maximum correlation would be 1. That's not true. There's no perfect correlation between a parent and their offspring, but there's a 0.5 about correlation between parent and offspring. Okay, suppose that's the only fact we know. Does that tell us? What does that tell us? Does that tell us that that the differences between a smarter kid and a dumber kid are due to genes or environment? Does it tell us anything about that? Can we make any inferences from that alone? No. Why?
Because they're being brought up by their parents.
But that's right, because the parents are providing both the genes and the environment. Right. So no, it tells us nothing. All right. Well, let's complicate life a little bit. Suppose we now look at both the data, the information we just looked at, which is the biological parent and child, but also compare an adoptive parent and an adopted child, where the child was adopted, within days of birth. Okay. This correlation, as we just said, is about a half. This one is about only half of that, which is considerably and certainly statistically reliably smaller. Okay. What can we now say? Does this change the picture? Yes. What does it say?
Genes play a bigger role.
Genes play a bigger role. The fact that there's this correlation in these biologically unrelated individuals means that obviously environment does play a role, right? But the fact that there's a much higher one here strongly suggests that that genes play a role too. Now, when I took the division of the class, about half of you thought it was mostly environment, and half of you thought it was mostly genes. But I dare say, if I said, is it all genes or all environment, probably you would have voted a different way. But there are a lot of people around who absolutely don't want to believe that this difference is for genetic reasons. I'll get to this second. There are lots of people who do not want to believe that differences in IQ have any genetic basis in a normal human population, nobody's brain damage or anything like that, have any impact on the differences in genes, have any impact on cause any difference in IQ. I'll go on with it. And so the question is, how do you wiggle out of it if you don't like the conclusion? Go ahead. What were you going to say?
Are there kind of between siblings who have like the same?
There are. We'll look at some sibling and some identical twins, fraternal twins, which comes to the same thing. Genetically comes to the same thing as sibling data in a minute. So yes, but this is it. Yeah. So suppose you don't like. Suppose you are an extreme environmentalist, and you don't like the claim which is. The obvious claim from this, a straightforward claim, you just don't think about it much, is that obviously this, as you said, this data suggests that there's a genetic component. But suppose you don't like that. You say, I just don't believe it. How can you wiggle out of it from this data? Can you think of some possible explanation that would explain this data, even though it's all, in reality, the differences are all environmental? I'm not going to ask for the answer now. I am going to ask for the answer at the beginning of next time, so think about it. Okay? I'll give you a hint. You may consider, I've only been able to think of one answer, and that's usually the answer that a few people in the class come up with, and it's not an answer that I think is highly plausible, but it is an answer that I think is possible. So think of if you don't want to believe that there's a genetic component, what could you say that would how could you accept this data and say, yeah, this data is right, but the conclusion that there's a genetic component is wrong? How could that be? So try to figure something out if you can. And that said, let's look at some other data. Actually, wait, this may not be quite well enough. No, this is not quite it. Well, your kind of data we'll talk about. Fraternal twins we'll talk about, or siblings we'll talk about in a few minutes. This is a different kind of data. It sometimes happens, not very often, but sometimes happens that first of all, identical twins. You have two kinds of twins, identical twins and fraternal twins. Identical twins literally are clones of each other. They have exactly the same gene. Okay? Fraternal twins are just like any other brother and sister. Okay? They're brother, sister, sister, brother, sister. They're just like ordinary siblings. So they have 50% of their commonality. What that 50% means, I'll talk more about in a little bit. But they're not genetically identical. They're just similar. Okay, if you compare identical, and sometimes happens, right? It sometimes happens that identical twins get separated at birth and are brought up in two different families for various reasons. And so people have made careers out of studying early separated identical twins because it's sort of interesting. Now you've got two individuals that are genetically identical, and one's raised by this family in one city, and this other one by some other family in some other city. To see similarities between them kind of suggests that something genetic is responsible. So, in fact, if you look at IQ correlations, the correlation for identical twins reared apart is very high. It's 0.8. There's quibbling about how high it is. There's some of the data upon which that number at least used to be based from a guy named Cyril Burt, who was eventually discovered to have been cheating a bit on his data analysis. Now, if you look at what he actually did, it wasn't really that bad, but nevertheless, it was not. It might have been influencing things. But there's lots of other data independent of this. This number varies anywhere from 0.7, 0.9 in different studies, but it's always pretty high. Unrelated children reared together in this set of data was 0.38, much, much, much lower. Okay, so this number here again says there's an environmental component to similarity. Environmental similarity produces IQ similarity, but here this is a huge, huge effect that looks like it's entirely genetic. I think this is a good time to stop. But before I do it, I also want to know if you can think of a way of wiggling out of that one if you are a strict environmentalist, or if you were a strict, even if you're not. Suppose you were a strict environmentalist. What could you think of to get out of that one?